Hello, everyone. I'm Ekaterina Bachkova, and I'm a part of ISTC team. Our project is dedicated to zoonotic tuberculosis. Today, I'm going to speak about prevention and control methods on farms and in domestic settings in order to overcome bovine tuberculosis. And now a few words about the causative agent of bovine tuberculosis. Mycobacterium bovis is a causative agent of bovine tuberculosis, and it is a member of the Mycobacterium tuberculosis complex. This complex also includes such important human pathogens as Mycobacterium tuberculosis. Also, it includes several other species, and they are phylogenetically very closely related to each other and share more than 99% chromosomal identity. Actually, they all cause tuberculosis with similar pathology in various mammalian hosts. Historically, taxonomic segregation of Mycobacterium tuberculosis complex has been based on each species' unique combination of host preference and its characteristic growth. And also morphology, physiology, and biochemistry were important features. Not to forget mentioning such important pathogen as Mycobacterium avium, which causes tuberculosis in birds, as well as in hogs and cattle. Mycobacterium bovis has a bacillar morphology. These microorganisms are rod-shaped, can also appear as cocci or filaments. Their size is 1 to 4 micrometer long and 0.2, 0.3 micrometer in diameter. In culture, they sometimes can form filaments up to 8 micrometer long. This microbe is facultative intracellular, obligate aerobic, acid-resistant, non-motile, and does not create spores. Its doubling time is from 15 to 20 hours. No other microorganism has as wide host range as Mycobacterium bovis. It can infect all warm-blooded vertebrates. Mycobacterium tuberculosis primarily affects humans, but also can be transmitted to hogs, cattle, and dogs. What is for M. bovis? It can affect swine, bison, deer and elk, camelids, llamas and alpacas, dogs and cats. Also, it can be transmitted to bisons, buffaloes, and which is the most important to us, cattle. Badgers can play an important role in transmission of bovine tuberculosis because they are considered to be a natural reservoir of Mycobacterium bovis, at least in some of the areas. M. bovis can survive in the environment, particularly in conditions that are chilly, gloomy, and damp. The microbe's survival period varies according to sunlight exposure, ranging from 18 to 332 days at 12 degrees to 24 degrees Celsius. According to research, bovine tuberculosis is still prevalent in underdeveloped and developing nations. Importantly, M. bovis is thought to be a causative agent of 10 to 15% of human tuberculosis cases. Thus, prevention of M. bovis spreading is very important for the eradication of tuberculosis in humans as well. Mycobacterium bovis is slow-growing microbe. That is why it might take months to detect the first signs of the disease in cattle. Various strains of Mycobacterium can cause different symptoms depending on where the infection is taking place. If the infection is in the gastrointestinal tract, A main symptom is extreme abdominal discomfort. An infection in the lungs causes severe cough. According to statistics, most tuberculosis lesions in cattle are found in the respiratory systems, lymph nodes, and in lungs. When present, clinical signs of TB can include variable pyrexia, weakness, anorexia, diarrhea, fluctuating temperature, loss of weight, enlargement of lymph nodes, and coughing, particularly when advanced TB takes place. These signs are not unique to bovine tuberculosis. Although normally TB shows a chronic deliberating disease, bovine tuberculosis can assume a more acute, rapidly progressive course. In the later stages of the disease, gradual emaciation with fever, weakness, and irregular appetite are typical signs. If the cattle is present outside in cold weather in the morning, animals suffering from TB typically have a more severe cough and may even get tachypnea or dyspnea. The advanced stages are characterized by serious respiratory problems and malnutrition. Animals become disoriented, unable to climb, and observed roaming around during the day. And now let's discuss various methods of TB diagnostics in cattle. So on this slide you can see what methods are currently used in both developed and developing countries, at least some of them. And one of the most common method is intradermal tuberculin test. This method is somehow similar to that of the human method, but of course it has some modifications related to the species and to their physiology. This method is widely used to prevent the spread among animals and to protect the public health as well. Like in humans, the test is conducted by intradermally injecting tuberculin, which triggers an immune response in infected individuals. Procedure for conducting the test in animals is as follows. The first stage is preparation. The test is usually performed on the neck or at the base of the tail in cows, where it is easier to assess the reaction. Second stage is tuberculin injection. A specific amount of tuberculin, specially formulated for animals, so-called PDD bovine, purified protein derivate from M. bovis, is injected under the skin of the animal. The third stage is waiting and evaluating the reaction. As with the human test, the injection site is examined from 44 to 72 hours later to measure any reaction in the form of swelling. If the animal is infected, inflammation and induration develop at the injection site. And finally, the fourth stage is evaluating the result. And here we can get negative or positive result. Negative result is the absence of a significant reaction, or in duration is smaller than the set threshold. It suggests that animal is likely not infected. Positive result is developing of significant in duration. It may indicate the presence of any infection. In this case, further testing and possible quarantine of the animals are required to prevent the spread of the disease. Also, not to forget mentioning that sometimes specific features can appear. For example, double tuberculin test can be carried out for more accurate diagnosis in animals. First, tuberculin specific to cattle, aforementioned PDD bovine, is injected, and it is followed by injection of human tuberculin, PPD, or of animal or avian tuberculin, PPD avian. Comparing the reaction to both types of tuberculin helps to differentiate infection of M. bovis from other mycobacteria. Actually, some factors can affect their results. Vaccination against tuberculosis in animals is not common practice, unlike in humans, so false positive results due to vaccination are less likely. However, stress or poor nutrition or immunodeficiency may weaken the response to the test, causing false negative results. Another method of diagnostics is histopathology. In this method, the post-mortem diagnostics of cattle suspected of being infected with M. bovis is carried out in the laboratory. This method includes inspection and, if needed, staining of the lymph nodes, particularly those connected to the head, chest, and belly. The presence of multiple granulomas in various organs is an indicator of severe pathological alterations. These granulomas typically have a yellowish hue and are caseous or calcified and are frequently encapsulated. Also, the presumptive diagnosis of mycobacteriosis can be made with hematoxylin and eosin staining if the tissue has characteristic histological lesions representing the animal's immune response to the infection. Caseous necrosis, mineralization, epithelioid cells, multinucleated giant cells, and macrophages. Next method of diagnostics is acid-fast staining. This is a common practice, which is considered to be one of the golden standards. The presence of mycobacteria in a given sample can be assessed by Ziehl-Neelsen staining. The smear samples are put on the microscopy slide, air-dried, fixed with fire, and stained according to a certain scheme. And then these samples are examined using the light microscope. The method itself is based on the structure of Mycobacterium cell wall. The cell wall is rigid and enables cells to retain the fuchsin-heated material after treatment for alcohol acid. In this type of coloring, alcohol acid-resistant microorganisms, including Mycobacteria, are pink-colored, while other microbial cells in the sample are stained blue. Thus, mycobacterial cells could be easily distinguished microscopically. Besides mycobacteria, other microorganisms are also acid resistant, such as Nocardia, Rhodococcus, and Corynebacterium, and they also can appear in the sample and can be stained with red or pink color. The advantages of this method of detection are that they are not as time-consuming as culture methods and not as expensive as molecular methods of detection. Also, these methods have some drawbacks as well. This method has a low sensitivity. Unfortunately, the presence of cells with the same acid properties as mycobacteria can take place. Also, in some cases, the sample may have a low concentration of acid-resistant cells, which can be missing while observing the sample under the microscope. Another staining technique was developed to overcome those drawbacks. It is considered to be more sensitive and specific. In this method, Oramin O is applied, and fluorescence microscopy is used for the same examination. The second big group of methods of detection is culture methods. Isolation of M. bovis is considered to be the golden standard for bovine tuberculosis diagnosis. However, this method has several disadvantages, which will be discussed later. Culture methods can be applied to isolate M. bovis from tissue or milk samples. In this method, different types of media are used. So the main advantage of this method is its high specificity. M. bovis can be differentiated from other members of Mycobacterium genus. Also, obtaining a pure culture of Mycobacterium allows further analysis of bacteria, which can be carried out using acid fast staining, various molecular methods, etc. However, this method has some serious disadvantages. And the first one is a long time period required for isolation and identification. In order not to get false positive results, the duration of culturing on solid medium should be no less than 8 weeks. Also, difficulties in obtaining samples of good quality may take place, and decontamination should appear, which is needed to avoid the presence of other unwanted microorganisms. However, decontamination can affect M. bovis as well and can cause various toxic effects on mycobacterial cells. Moreover, the obtained results could be false negative due to the actually low concentration of mycobacterial cells in the sample. This is why a combination of various methods of diagnostics is required. It is difficult to distinguish M. bovis from similar species, so molecular diagnostics methods should be applied along with microbiological methods. Isolates of M. bovis can be obtained on liquid or on solid media. Also, a combination of both types media can be used. Solid media can be either egg-based or agar-based. Slow growth of M. bovis is one of the main limiting factors affecting rapid detection of TB. The first colonies appear no earlier than 7 days, and the most reliable results should be achieved only after several weeks of cultivation. The duration also depends on concentration of the pathogen cells in the tissues. To enhance the growth of the colonies, pure watt is added to the medium. It is not clear which type of the medium, solid or liquid, demonstrates the best results for M. bovis. Thus, both types of medium are widely used. The following steps are essential when culture method of M. bovis detection is applied. The first is collecting samples. In most cases, cotton nasal swabs are used to collect samples as they are believed to improve sensitivity and reduce the risk of contamination. If the tissues are used, cutting out the affected areas with TB-like lesions is needed. The second stage is homogenization of the sample, which is followed by decontamination. It is the special procedure used to avoid the presence of other microorganisms. Usually, decontamination involves the addition of various chemical compounds and incubation for several minutes. Next stage is seeding, which is spreading the pretreated sample on the slant agar media or on liquid media. Of course, sample pretreatment and seeding should be carried out in sterile conditions in the special laboratory box. The next stage is incubation. Seeded tubes should be kept under 37 degrees Celsius in a thermostat. They should be examined daily during the first week and then once a week for the period of 10 to 12 weeks. During these examinations, all changes which have occurred with the seeding are noted, whether the media changed its color or if the colonies have appeared. Characteristic growth patterns and colonial morphology can provide a presumptive diagnosis of M. bovis presence. However, every isolate needs to be confirmed. And it is, of course, very important to distinguish M. bovis from the other members of M. tuberculosis complex.
Hello again, I'm Katerina Bachkova, and now I'll perform seeding of TB-like lesions on special media for culturing mycobacteria. Here is our sample, which contains TB-like lesions. And now we'll proceed it with the phenol red method with subsequent seeding. First, we should measure 7.5 milliliters of phenol red solution. All the procedures are done under the protective laboratory hood or in a laboratory box. It should be equipped with special filters and certified for working with tuberculosis. In this test tube we have 7.5 milliliter of phenol red solution. Now it's time to turn on the flame. We take our sample using the sterile instruments to put it in on the Petri dish. All the TB-like lesions and capsules which look like tuberculosis should be cut out using the scissors. Also, we can search for TB-like lesions inside those tissues. The outer layer of the tissues is of a special interest because usually it contains most of the mycobacterial cells. From time to time, we should sterilize our scissors and flame. here we have the cut out pieces of the sample. We should put them in this mortar. The excessive tissue is placed, is returned to the tube. The instruments can be put into the bleach, and now we take our phenol red solution, add some drops of phenol red, and here we have sterilized sand. Add some sand and start grinding the sample. This procedure should be carried out at least 10 minutes. After grinding, we should take 7.5 milliliters of phenol red with the grinded sample and put it into the new test tube. So by now we see that our sample is perfectly grinded. We take the sterile pipette, open the test tube, now the sample is carefully collected. Now we take 2.5 milliliters of natrium hydroxide, one normal, and place it into the same tube. Now again we have 10 minutes incubation. Addition of natrium hydroxide is done for decontamination. So after incubation of our sample with natrium hydroxide, after decontamination stage, we should add sodium chloride. Now we continue our sealing procedure. After centrifugation, we take the test tube from the centrifuge and place it onto the laboratory box together with the two test tubes for seeding. Here in these test tubes we have two types of media. Here Colexus medium and here Löwenstein-Jensen with pyruvat. We labeled both test tubes. We signed them with the number of normally the 1 milliliter is sample. And also here we have the date of seeding and orange. Type of pretreatment, phenol red. Turn on the flame. Open the disposal box. And we remove the supernatant. Our test tube from the protective laboratory box into the centrifuge and centrifuge it at 3000 G for 25 minutes. Now we place our test tube to the centrifuge. First we'll inoculate the test tube with Löwenstein-Jensen medium. We put it directly to the flame, open the test tube. We need one sterile loop and our test tube after centrifugation. We open the test tube. Take small amount of the sample, close, and now we apply 3000 on the slant egg and centrifuge for 25 minutes. Now Now
the loop
our
is discarded
seeding procedure after
and the
centrifugation.
test tube is closed
The test tube.
from the centrifuge. We'll perform the same again for the second type of
Place
media
it onto the laboratory box.
together with the two test tubes for seeding. Here in these test tubes we have two types of media. Here Colexis medium and here Löwenstein-Jensen with pyruvat. We labeled both test tubes. We signed them with the number of the sample. Also here we have the date of seeding and type of pretreatment, phenol red. This test tube is not needed anymore, so we'll open the disposal box and we remove the supernatant. Close the disposal box. Remove the second pair of gloves. Now the test tubes are taken from the box. And they should be placed. First, we'll inoculate the test tube with Löwenstein-Jensen medium. For the first week, we put it directly to the flame.
Under the angle.
Open the test tube. We need one sterile loop and our test tube afterwards centrifugation.
They are stored vertically.
We open the test tube.
The samples are examined every day during the first week of incubation, and after the first week,
Take
they
small
are
amount
examined
of
weekly.
the sample.
The incubation usually takes from 10 to 12 weeks.
Close. And now we apply on the slant agar. Now the loop is discarded and the test tube is closed. We'll perform the same again for the second type of media. This test tube is not needed anymore, so we'll discard it. Turn off the flame. Close the disposal box. Remove the second pair of gloves. Now the test tubes are taken from the box. And they should be placed into the thermostat to be kept under 37 degrees Celsius. For the first week, the test tube are stored under the angle. And afterwards, they are stored vertically. The samples are examined every day during the first week of incubation,
and after the first week they are examined weekly. The incubation usually takes from 10 to 12 weeks.
The next method is involved in the group of molecular biology methods. It is a well-known polymerase chain reaction, or a so-called PCR. PCR includes isolation of DNA of samples and subsequent reaction of polymerization, which is accompanied by analysis and interpretation of the results. While culture methods still remain a golden standard for TB diagnostics, exceptionally long culturing period postpones important decisions and suspected disease outbreaks. So the urge for rapid tests has been pressing. On the other hand, a task of differentiating between various members of MTB complex is not a simple one and would usually require a combination of slow, labor-intensive, and time-consuming tests, whose results are often not reproducible. This is where PCR has proven to be a good solution, and this is why it has been successfully applied to discern members of Mycobacterium complex and to directly detect M. bovis in bovine tissue samples. Advantages of this method is, of course, a possibility of rapid detection and high specificity. However, there are certain disadvantages as well. This method requires special equipment, which can be used only in laboratory of a high standard. Actually, no standard for PCR of Mycobacterium bovis exists, and different available commercial tests involve various specificity and sensitivity. And interpretation of results in this case can be challenging. Also, DNA isolation can be a difficult procedure because sometimes there are low concentrations of M. bovis cells in the tissue samples, and due to their complicated cell wall structure, DNA isolation can be difficult. Currently, there are several commercially available diagnostics kits available for detecting of M. bovis. Some of them can be seen on this slide. And the next large group of methods is serological tests. And the first one, which I would like to tell, is so-called ELISA, enzyme-linked immunosorbent analysis. It is a method of TB diagnostics in cattle, which has a growing popularity. The method is based on the highly specific reaction that exists in the organism between the antigen and antibody. In Mycobacterium bovis, the purified antigens are used, such as AG85 antigen, which comprises the majority of secreted proteins. Also, MPB70 and MPB83 proteins are used. The main advantages of this method is its simplicity and low cost and low logistical demands. The samples for detection can be stored before the analysis is carried out, which is very important in the laboratory practice. Their sensitivity, however, is lower than in other tests, particularly in recently infected animals, increasing in advanced stages of the disease. The use of combinations of specific antigens has been demonstrated to increase the sensitivity and specificity of serological tests. ELISA has been proposed as a useful addition to other methods of diagnostics, and it is especially important for TB detection in wildlife and as available additive to tuberculin skin test. Also, we have another method of diagnostics, which is also included into serological tests. It is IGRA, interferon gamma release assays. In this method, this method is based on the fact that immune system cells infected with M. bovis release interferon gamma in response to specific antigens. These antigens are not present in the mycobacteria used in vaccine, making IGRA more specific for diagnosis active or latent tuberculosis. The main advantages of this method are its specificity, safety, and convenience. The method does not require a follow-up visit to check for a reaction, as with the tuberculin test, since the entire process is based on a single blood draw. Also, this method is more accurate diagnosis of latent infection, which may be harder to detect with some other methods. However, there are certain limitations of IGRA. It is rather expensive and complicated method and requires some special equipment. Also, even though IGRA are more specific, they may not be sensitive enough in some cases, particularly if the animal is in the late stages of the disease or has a weakened immune system. In this case, the reaction cannot be developed in the organism and cannot be detected using this method. Also, there is limited data on use in different species. While IGRAs are widely used for diagnosis tuberculosis in humans, their use in animals, including cattle, is still being studied and implemented in some regions. IGRAs are often used in combination with some diagnostics methods, such as in tuberculin skin test or molecular methods, and provide a more complete picture of the herd's infectious status. A combined approach helps to improve the accuracy of this method and to reduce the risk of tuberculosis spreading among animals and humans. So the best results could be achieved when using a combination of various methods of detection of M. bovis. In this case, the diagnostics would be more accurate. The prevention and control of bovine tuberculosis is very important because sick animals lose productivity and also because humans are at risk of contracting the disease. However, control efforts are not implemented or are not implemented well enough in the majority of developing countries. The reasons are connected with the following factors. Firstly, it is financial constraints. TB prevention activity requires a lot of money, which could not be present in developing economies. Secondly, some of the developing countries lack proper legislative regulation and control mechanisms to prevent spreading of TB. Moreover, in some countries, a lack of skilled professionals is observed, along with the low assessment of the importance of zoonotic tuberculosis in the animal and in public health sectors. Besides, for some nations, such control measures for bovine tuberculosis prevention as slaughterhouse inspections and culling of infected animals can be not only expensive, but unacceptable due to ethical or religious reasons. The following measures should be taken for prevention of B2B spreading. And to prevent person-to-person transmission, patients infected with M. bovis should be treated with the same public health procedures used to treat patients with infectious M. tuberculosis. Cattle with tuberculosis should not receive any treatment at all, and any affected livestock should be slaughtered. This is because M. bovis is resistant to pyrazinamide, a medication that is frequently used to treat M. tuberculosis complex-derived infections. It is actually necessary to provide specific hygiene standards for food of animal origin to stop diseased animals from getting into the food chain and lower the hazard connected with the consuming tainted milk or tainted meat. The first step is pasteurization of milk, which should become a mandatory procedure. Systems for inspecting meat need to be improved and intended to stop the general populations from consuming tainted goods. An examination must be performed both pre and post-mortem on any animal that enters the food chain. Thus, the slaughterhouse inspections should play a key role in these processes. Also, veterinarians are highly involved in B2B prevention activities. Thus, proper testing of cattle should become a routine procedure in all countries at risk of B2B. Such tests should be reliable, fast, and cheap. The tuberculin test is the primary method for detecting bovine tuberculosis in cows. A tuberculin test needs to be done on cattle 12 months before slaughter. The early detection of preclinical infection in animals meant for food production and the prompt removal of diseased animals from the herd will prevent future sources of infection for people and other animals, making tuberculin testing helpful in the control of bovine tuberculosis. Vaccination for the prevention of bovine tuberculosis is another option that is being investigated. The vaccine reduces the severity of the disease, usually allowing the bacteria to infect only a few lymph nodes, but does not prevent infection. The goal is to develop an enhanced vaccine that can provide protection against the disease. The vaccine should be given to wildlife in an effort to prevent the spread of the disease to cattle. It is worth mentioning that in some cases it is found reasonably to isolate the cattle from the wild animals, first of all from deer, that can be infected with M. bovis as well. Also, some of the bacterial cells can survive for a long time on the deer saliva. Thus, it is important to prevent contacts with the saliva remains. On the major concerns regarding bovine tuberculosis is the possibility of its transmission to humans. In Georgia, tuberculosis remains an important public health problem. Despite significant progress in the implementation of TB control interventions during the last decade, the country faces a number of important challenges that require further intensified and streamlined efforts to effectively control the epidemic by securing universal access to prevention, diagnosis, and treatment of all forms of the disease, and ensuring effective integration and management of TB control interventions within the overall health system framework. The government of Georgia is committed to increasingly allocate financial and other resources for effective TB control. The legal framework for TB control in Georgia exists. This is a set of particular laws and regulations which stipulate the right of Georgian citizens to receive quality medical services. TB control services in Georgia are provided by both public and private health care institutions. TB laboratories have been downsized and integrated into the framework of public health system under the management of the National Center for Disease Control and Public Health. TB care delivery in Georgia is integrated into the provision of general health care systems. TB services are provided by specialized TB units as well as by primary healthcare units, PHC. TB detection is mainly done through passive case finding. The country uses a standardized TB recording and reporting system, which has been upgraded to include the latest World Health Organization recommendations and additional country needs. Individualized recording and reporting is incorporated into the National Electronic TB Database. This strategy places particular emphasis on implementation of the integrated TB electronic database, which is one of the main intervention areas. The overall goal of TB control in Georgia is to decrease the burden of TB and its impact over the overall social and economic development in the country by ensuring universal access to timely and quality diagnosis and treatment of all forms of tuberculosis, which will decrease illness and deaths and prevent further development of drug resistance. To sum it up, we can say that Mycobacterium bovis is the main causative agent of bovine tuberculosis, a member of Mycobacterium group, which has a wide host range. It affects various mammals, including human. A variety of methods for the detection of bovine tuberculosis exist. All methods have their pros and cons. For the better results, a combination of different methods should be applied. Veterinary services play a key role in prevention and detection of bovine tuberculosis cases. This is accomplished through routine testing, screening for the sick animals, and introducing vaccination and proper hygiene standards and regulations. In Georgia, a national program for tuberculosis prevention exists. This program involves all forms of tuberculosis with the main aim to reduce the number of TB cases both in humans and in cattle and to prevent the further development of drug-resistant bacteria. On this slide, you can see some of the references which were used to make this presentation. They include useful information on methods of bovine tuberculosis detection. And that's all. Thank you for the attention. Goodbye.